Have you ever wondered how our eyes and vision evolved to become so intricate? Well, you have reached the right video to explore this question. Before we get into the mechanics of the eye, let's begin with what and how our eyes perceive objects in our visual field. Eyes perceive light, which is a form of electromagnetic radiation. Us humans specifically process the visible light spectrum. The light reflects off of objects in our visual field, and the frequency and amplitude of the light wave will determine the color and brightness that is perceived. When light energy reaches our eyes, it travels to the back of our eye to a thin layer of tissue known as the retina. The retina is composed of various types of cells, but the light is first processed by the posterior layer of the retina, the photoreceptors. Photoreceptors transduce light energy into nerve impulses, which is then sent to the brain to be processed by the rest of the visual system. In fact, there are two types of photoreceptors, rods and cones. Rods detect black and white and are spread mostly in the periphery of the retina, whereas cones are concentrated in the center of the retina and detect high resolution and color. Clearly, we have these very specialized structures to process light, but how did vision evolve to become as complex as it is? This question has perplexed many biologists, even Charles Darwin himself. Well, over 500 million years ago, the simplest version of the eye can be traced back to unicellular organisms like euglena. They possess a structure known as eye spots located at the base of the flagellum. This structure is composed of a patch of photoreceptors, and it functions to transduce light energy to trigger flagellar and cell movements. Having this ability to sense brightness and distinguish between light and dark provided an evolutionary advantage to these organisms. The eye spot promotes movement towards light to facilitate the process of photosynthesis and thereby aiding with survival. While the eye spots do permit the ability to sense the presence and intensity of light, they do not permit the ability to sense directional information of light. On a flat eye spot, the same patch of cells may be activated regardless of the angle of the incoming light ray. Therefore, the flat eye patch arose into a pitted cup shape in the Cambrian period, with a photoreceptor layer, a pigmented cell outer layer, and a single opening for light. Now, incoming light rays at different angles would excite different patches and thus provide directional information. Organisms like the planaria possess this pit eye. The pit eye provided an evolutionary advantage as it facilitates the ability to discern direction, distinguish moving objects like predators or food from the surroundings, and permit them to seek out shade to hide from predators, which, put all together, increase the chances of survival and reproduction compared to animals with eye spots. After the pit eye, there were even more biological changes that led to improvements in the vision. We can especially see this in the nautilus during the Cambrian explosion. The primitive nautilus eye functions like a pinhole camera. The overall structure of the pit eye is retained, however the light opening structure in the pit eye is reduced to enhance visual acuity. Upon entering, fewer light rays interfere and thus provide a higher resolution image. However, a smaller opening presented a trade-off between resolution and brightness, since a smaller opening meant fewer rays could enter and created a dim image. The improved vision provided an evolutionary advantage in the deep, dark, and murky waters, by enabling them to capture and locate prey and permitted them to become a successful, fast-acting predatory group. Next, the pinhole camera eye developed a cornea or transparent cells to cover the opening of the eye to prevent infections, like we see in Onychophora during the Cenomanian age. This covering further allowed the inside of the eye to fill with a fluid known as the transparent humor, which not only provided structural integrity to the eye shape, but also assists with the refraction of light onto the retina for light processing. After, crystalline proteins formed the lens, which further optimized the refraction and focusing of light on a specific point on the retina. The lens, in conjunction with the ciliary body, significantly assisted with the focusing abilities on near and far objects. This overall refined camera-like eye is clearly seen in squids or octopi, which provided them a huge survival advantage in their specific environment, particularly to see great in the deep dark water, watch for threatening whale predators, and locate prey. Further refinements to this camera eye would further develop it into a human eye. These refinements included photoreceptors increasing in number to provide more detailed images, the iris, which is your eye color and is responsible for controlling the diameter and the size of the pupil, therefore controlling how much light enters the retina to ensure a clear image, and the brain. The coevolution of the brain was also a significant part in the evolution of the eye as well, since the visual cortex was able to process sharper images. This cladogram is a great summary and overview of my presentation and captures how a simple eye evolved into a camera-style human eye. Thank you for listening to my presentation.